A very good evening, everyone. Welcome to today's class, and we are going to crack GATE 2021 together. And you're watching this video on Let's Crack CSIR UGC NET. Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET, GATE examination, and various other examinations. And India's best educators they teach on Unacademy. The topic of today is cellular totipotency in plants. But before beginning with the class, let us have a small introduction about myself and Unacademy. I am your educator Farida Bandookwala and I have a teaching experience of 10 years. I have qualified MP SLET examination in year 2018. If you're opting for Unacademy, what all things you are going to get? You are going to get daily life classes so that you will be always in that momentum of studies and you can ask your doubts and queries directly with the faculty members. The whole syllabus is quite structured. That is the 13 units of CSIR you can cover in a small span of time, and for GATE also a crash course is going on. So you can complete the whole thing in just a span of 1.5 months. Live tests and quizzes are conducted regularly so that you can evaluate that how much you have learned so far, and unlimited access of all the videos which are recorded under the category of CSIR UGC NET or whichever category you are opting for. Unacademy tries to give as many free live classes as it can. So the classes are conducted on YouTube and there are special classes. So these two are the kind of free classes. For YouTube class, you just need to subscribe the channel and come whenever the class is live, or you can watch the recorded class as well. For special class, what you need to do is you need to download the Unacademy Learning app from Google Play Store. Then you can make your account, and after making account, you can use my referral code FB01 for accessing the special free classes. So by using my referral code FB01, you can watch my special classes as well as the special classes done by other educators. So you can watch my special classes or the special classes of other educators which is live or which were recorded in past. Anything can be done. Then there is a plus subscription. This plus subscription is a paid subscription, but it is a very pocket friendly subscription and very helpful to qualify the examination, because for all the examination short term courses, crash courses are conducted, and all the 13 units of CSIR and all the units of GATE is actually covered in the plus subscription, and you can get the whole study material as well. Recently, a discount is going on for the plus subscription, and this discount is going to end soon. So go for the plus subscription. For 12 months, it is going to cost you 11,550 rupees, and for 24 months, it is going to cost you 16,800 rupees. And the story is not yet over. You can get a 10% discount by using my referral code FB01. So do not forget to use my referral code FB01, and for 12 months the course is going to cost you 10,395 rupees, and for 24 months it is going to cost you 15,120 rupees. Soon the prices are going to increase. There is after 31st of December, the prices are actually going to increase. That is the 31st December 2020. So just go for the subscription, and by getting just one subscription, you can watch the videos of all the top educators. You can attempt the quizzes, you can attempt the mock tests, you can attempt various courses which are taken in this category. Then you can attempt the various batches, and you can download the study material in PDF format. So these all things you are going to get by a single subscription. So do not wait and go for the subscription. By one subscription, you can watch the videos of other educators as well. So a single topic is actually taken by so many faculty members. Whichever thing you like the most, you can go for it. So this was a small introduction about Unacademy. Let us start with the topic, that is sorry. So that is the cellular totipotency in plants. Cellular totipotency in plants. Let us begin this with the introduction. Then we'll be talking about various things under this topic. So for introduction, so what is totipotency? First of all, we should know what is the meaning of totipotency. Totipotency is the ability of a single cell to divide and produce all the differentiated cells in an organism. Ability of a single cell, ability of a single cell to divide and produce all of the differentiated cells in an organism. In an organism. This is the definition of totipotency. That from a single cell, the whole organism can be generated. All the differentiated cells in the body can be made. So that is the definition. And in other words, totipotency is the genetic potential of a cell to produce the entire organism. So when we are talking in terms of plants, so what are we going to call it in terms of plants? It's a genetic potential. It's a genetic potential. The genetic potential of a plant cell to produce entire plant. To produce entire plant. This is the totipotency, genetic potential. All the genes would be working in a direction to produce the whole plant. Now, isolated cells from the differentiated tissues are generally non-dividing and they are quiescent or they express certain genes. To express totipotency, they need to undergo dedifferentiation and then redifferentiation. So, differentiated tissues which are actually non-dividing or which are actually quiescent, they need to undergo for becoming totipotent, what they need to do? They need to undergo dedifferentiation, and then it needs to undergo redifferentiation. This is the process. So in Latin word or in Latin language, totus means entirely, and potens means having power. So this is the word totipotency came into being. Totipotency. So this word totipotency is being taken from Latin language. Latin words. Totus and potens. Totus means entirely, potens means having power. So that is how the word totipotency was generated. So the possibility of regenerating an entire plant from a single or few non-zygotic cells was actually proposed by a scientist called as Herbert Land in year 1902. So Herbert Lee is also popularly known as father of tissue culture. So Herbert Land in year 1902 gave the concept of regenerating entire plant from single from single or few non-zygotic plants. Non-zygotic cells, not plants. So Haberland is also called as father of tissue culture. Remember this. Haberland is called as father of tissue culture. So how this whole thing is going to actually occur, that we will see that how in culture systems various like callus, suspension and other things, how it is going to happen. So first of all, we'll be taking a totipotent cell. This is a flow chart. We'll be taking a totipotent cells. This totipotent cell, how it is generated, maybe a differentiated cell was taken, then it was de-differentiated, and then totipotent cell was obtained. So after totipotent cell was obtained, then differentiation was done. Differentiation of the totipotent plant cell was done. How differentiation would be done? By using various different kind of hormones. Then embryogenesis can be done with the undifferentiated plants. So differentiation in the form of embryogenesis, in the form of organogenesis can be done. Then it can be actually taken into histogenesis. Histogenesis or cytodifferentiation or cytodifferentiation. Embryogenesis is going to lead into embryoid. Then embryoid is going to lead into complete plant. Through organogenesis, various things can be done, such as collagenesis. Collagenesis is going to lead into only shoots. Remember, collagenesis is only for shoots. Then rhizogenesis is going to lead into only roots. And colorhizogenesis. Colorhizogenesis is going to lead into shoots with roots. And here this histogenesis or cytodifferentiation is actually leading into production of xylem and phloem. So this is how the process of various things which can be obtained from totipotent cells can be done. So high ratio of cytokinin to auxin, high ratio of cytokinin to auxin is going into shoot development or collagenesis, and cytokinin to auxin low ratio is going to lead into root development, that is rhizogenesis. So these two things you should remember. I hope the thing is quite clear. Now talking about culture systems. Culture systems. So first culture system that we are going to see is callus culture. So callus, what is a callus? First of all, let us talk about callus. Callus is unspecialized, unorganized, growing and dividing mass of cell. Growing and dividing mass of cells. It is unspecialized, unorganized, and it is into live state. It can grow and it can divide. Then it can be maintained indefinitely. After that, it is suitable for cytodifferentiation. suitable for cytodifferentiation. Then it does not perform any kind of photosynthesis and it grows in dark. No photosynthesis, grows in dark, grows in dark. So no photosynthesis, it is actually yellowish in color. It is yellowish in color. No chlorophyll. Can be used to isolate single totipotent cells. Can be used for getting single totipotent cells. Totipotent cells. Now many cultures, they lose their potential for differentiation during the continual subcultures due to epigenetic changes. Like if we get a callus and we just keep on reculturing, subculturing. So because of various of epigenetic changes, they might lose that capability of totipotency. Difficult to follow many cellular events during its growth and developmental phases can be the other drawback of callus culture. After callus culture, the second type of culture system that we can see is suspension culture. Suspension culture. So, suspension culture, the word itself is saying here, callus culture is kind of solid tissue grows, solid tissue or a mass of tissue. A mass of tissue is grown. Callus is a kind of mass of cells or mass of tissue. And in suspension, the type of culture in which single cells or small aggregate of cells multiply while suspended in agitated liquid. So in this, single cells or small aggregates of cells or small aggregates of cells multiply in suspension, in suspension, in agitated medium. The medium would be in continuous motion so that whenever new cell is formed, it just dislocates from the whole mass. So single cells are obtained and small aggregates are obtained. Suspension in agitated, liquid medium. After that, it receives more homogeneous stimuli in a defined medium, supplemented with requisite amount of inducers such as sugar and oxygen because it is in continuous motion. So receives more homogeneous stimuli. Receives more homogeneous stimuli such as sugars and hormones because of the continuous agitation. It can be very much suitable for the production of secondary metabolites such as alkaloids. So can be studied for the production of secondary metabolites such as alkaloids. Then it is possible to analyze the biochemical pathways related to differentiation of cells in suspension culture. Can be used to analyze biochemical pathways related to differentiation of cells. So this was about suspension culture. Now talking about the single cell culture. Single cell culture. So single cell culture is a method of growing isolated single cells aseptically on nutrient medium under controlled conditions. Used for growing single cell for single cells aseptically on a nutrient medium under controlled condition, obviously under controlled condition, and in all the conditions would be controlled, like previously the cultures which we have seen in that also the condition would be controlled condition. After that, can be isolated from a variety of tissues and organ of green plants, as well as from callus tissue, cell suspension, either mechanically or experimentally, enzymatically. So can be obtained from what all things can be obtained from callus tissue, from suspension culture, callus tissue, suspension culture can be obtained mechanically or enzymatically. And can be used successfully to get the single cell clones, can produce single cell clones, and it can be used to regenerate plants can be regenerated from the callus tissue derived from single cell clones. Whole plant can be regenerated. Whole plant regeneration. Then isolated single cells can be handled as a microbial system for the treatment of mutagens and mutant selection. And single cell culture is an ideal system for the study of biotransformation. For studying biotransformation. This is about single cell culture. Now we'll be talking about the protoplast cultures. So protoplast, what is a protoplast? Protoplast is a naked plant cells without the cell wall. So we have done a class on protoplast fusion also, right? And what are the uses of protoplast fusion and everything. We have done a class. You can go and watch the video. So protoplast, what is a protoplast? Protoplast is a naked plant cell. So we would not write that. We would write plant cell minus cell wall. Plant cell minus cell wall, but they have got plasma membrane and all the other cellular components. So plant cell minus cell wall is protoplast. This protoplast is isolated more frequently from mesophyll type of tissue, and the whole method of isolation we have covered in protoplast fusion. Isolated from mesophyll cells, oblique tissue of fully expanded leaves of young plants, new shoots, either mechanically or enzymatically. We have done this. Then protoplast culture can be regenerated into whole plant. Can be regenerated into whole plant. Then hybrids can also be developed from protoplast fusion. So hybrids, we have seen hybrids and hybrids. So hybrids and hybrids can be obtained from protoplast fusion. Hybrids and hybrids can be obtained through fusion. And it is easy to perform single cell cloning with protoplast. Single cell cell clones. Then genetic transformations can be achieved through genetic engineering of protoplast. It is easy to perform the genetic transformation in protoplast. Genetic transformation is easy in protoplast. Then protoplast is an excellent material for studying the ultrastructural studies. Ultrastructural studies. Ultrastructural studies. Then isolation of cell organelles and chromosomes is very easy with the help of protoplast cultures. Isolation of organelles and chromosomes is easy. Then protoplasts are also very much useful for membrane studies. Useful for membrane studies. We'll write here useful for membrane studies. That is, the transport and uptake processes can be very much easily studied with the help of protoplast culture. Now there are some factors which affect the totipotency. So we'll see that factors that affect cellular totipotency. So what are those? First is source of explant. What is the source of explant? Second thing, it will depend on nutrient media and their constituents. Nutrient media and constituents. Then it depends on culture environment, that is the physical form of medium. Culture environment, that is physical form of media. Physical form of medium. So let us talk about them in a very brief manner. So source of explant, organ that serve as a tissue source. Physiological and ontological age of organ is going to matter in the source of plant, and the season in which the explant is obtained is also going to affect the cellular totipotency. So overall, the quality of explant is going to affect the cellular totipotency. Now, nutrient media and constituents, inorganic, macro and micronutrients, carbon and energy source, and nitrogen source, and the type of hormone constituent you are using, that all is going to affect the cellular totipotency. Now, the physical form of medium, the presence and absence of agar, whether it's a liquid medium, suspension medium, what is the pH of the medium, what is the quality of light taken, what is the quantity of light provided, what is the temperature, relative humidity, gaseous atmosphere of the vessel. These all things are going to affect the culture in the cellular totipotency, overall in the cellular totipotency. So everything, that is the presence or absence of agar, or the pH of the medium, then the quantity and quality of light, quantity and quality of light, then temperature, humidity, gaseous environment of vessel, those all things are going to affect the cellular totipotency. Now when we are talking about totipotency, so are there any importance? What have I written? So is there any importance? So what is the importance of totipotency? Lastly, we'll be seeing this. So what is the importance of cellular potency? Importance of cellular totipotency in plants. So first is vegetative propagation can be done. Vegetative propagation. Any part of the plant can be taken and whole plant can be formed from there, that part. So vegetative propagation. Without any kind of variation, the clone of the whole plant can be made. So vegetative propagation. Then germplasm preservation, the whole germplasm can be preserved in the form of suspension culture, callus culture, that all thing. So it is highly useful for germplasm conservation. Germplasm conservation. Like imagine there's a plant which is unable to propagate on its own. So we can take a small part of that plant and develop it into a callus or through suspension culture and lead it into undifferentiated form and develop many copies of that plant. So that is also a germplasm conservation, not directly preserving it on the artificial media, but making clone of that plant with the help of its capability of cellular totipotency. Then various plants, they have a large seed dormancy. So this seed dormancy can be overcome by using some somatic cells of the plant to develop the whole plant. Then embryo rescue can also be the importance of cellular totipotency. Then protoplast fusion, we have seen the if we are obtaining the protoplast and then we are going for uh, uh, fusion of two different type of species of plants or two different strain of plants, and then we can develop a hybrid. So that is also possible just only because of cellular totipotency. That is not possible with any other species. That is not possible with humans. That is not possible with monkeys. Not possible with guinea pigs. This protoplast fusion only goes to cellular totipotency in plants, specifically in plants. Then haploidy can be attained with the help of cellular totipotency. Then genetic modifications are very much easier through this protoplast cultures. Genetic modifications are easy. Genetic modifications. Genetic modifications are easier. So like this, we have covered the topic cellular totipotency in plants in a decent manner. That is, we have tried to cover almost all the aspects which are related to this. So we'll just do a very quick revision that what all things we have done in this, right? So let us revise that what all things we have done. We have seen the definition, that is ability of a single cell to divide into all the cells of the plant, and that is the genetic potential of a plant cells, which is not the case with the human cells or any other animal species. So how this is done? Differentiated tissue is taken. This differentiated tissue is basically the explant. Explant is taken. Then it is actually in the non-dividing state or quiescent state. So it has to be changed into totipotent, and then for changing it into totipotent, it has to undergo dedifferentiation and then redifferentiation for developing the whole plant. After that, this totipotency word came from two words, that is totus and potens. Totus means entirely, potens means having the power. So it's a Latin word. And Hebertland is called as father of tissue culture. After that, we have seen how totipotent cell is responsible for so many things: embryogenesis, organogenesis, and histogenesis. So high ratio of cytokinin to auxin is going to lead into shoot formation, and low ratio of cytokinin to auxin is going to lead into root culture. Then culture systems we have seen. The first culture system that we have seen is callus culture. So callus is yellowish mass of cell. It is unspecialized. It can be maintained indefinitely so that it is useful for germplasm conservation. Germplasm conservation. It can be used for getting single totipotent cells. Second culture system we have seen is suspension culture. In this, the whole system is in continuous agitation form. So single cells are obtained. It is used for secondary metabolite studies such as alkaloids. And it is also useful for analyzing the biochemical pathways. Then single cell cultures are done. And the single cell cultures are used for growing single cells aseptically and very good for studying biotransformation. After that, we have seen protoplast culture. So protoplasts are obtained mechanically or enzymatically. It is very useful for studying the membrane studies. Very easy to do the genetic transformation in protoplast, and isolation of organelles and chromosomes can be very easily done in protoplast cultures. Then factors which affect the cellular totipotency, that is the source of explant, whether it is from a young plant, old plant, what is the season in which it is obtained, those all things are going to affect. Then nutrient, media and constituent, like which hormone composition you are using, with what is the carbon source, what is the glucose source, that is going to affect the cellular totipotency. Then what is the culture environment is going to affect. Then we have seen the importance that it is important in vegetative propagation, germplasm conservation, seed dormancy, embryo rescue, protoplast fusion, haploidy, genetic transformation, and much more of the things. So like this, we have completed this topic, that is cellular totipotency in plant cells. And as we say in the starting and end, let's crack CSIR UGC NET together. And you are watching this video under the category of GATE crash course for 2021. If you haven't subscribed our channel till now, subscribe the channel, hit the bell icon for getting all the notification. Like my video if really you have obtained something out of it. Use my referral code FB01 for getting additional discount of 10% in the plus subscription, and also you can use my referral code for this special classes. So my referral code is FB01. Do not forget to use my referral code FB01. So it's a thank you from my side. A very happy new year to everyone. In this new year, qualify all the examination which you are planning for, and make the best of your life. Keep on studying, and the another most important thing is to be happy. So just be happy and qualify all those examinations which you are planning to qualify. So it's a bye from my side. We'll be meeting tomorrow again at 10:30 with an interesting topic of CSIR syllabus, and in the evening 5 o'clock with the interesting topic of GATE syllabus. Bye. Write your queries, write any specific topic which you want to study in the comment box. Bye.